Very good morning, dear students. So we have started the next unit number 3 of biochemistry, and this unit is mainly deals with the most important type of biomolecule in the world, that is proteins. Okay. So we have already discussed the basic aspects of proteins in the previous class. So the next and first topic mentioned in this unit, amino acids and proteins and important functions of proteins. So first we will discuss amino acids, the basics and their properties of amino acid. Then briefly we will discuss about proteins. Then we will discuss a brief outline of the important function of the proteins. So amino acids are the building blocks of proteins. Okay. So each amino acid is a nitrogenous compound which have both an acidic carboxyl group, that is COOH group, and a basic amino or NH2 group. So this compound which have an NH2 group at one end and carboxylic acid group at the other end. Then they are having an R side chains. Okay, this R stands for the side chains that are different for each of the amino acids. And the R can be symbol as a hydrogen atom or a methyl group or a complex structure. And so how will you define an amino acid? So amino acid may be defined as these are alpha amino substituted carboxylic acids. So amino acids are alpha amino substituted carboxylic acid. So on the left side you can see the structural compartment, the general structure of an amino acid. Okay, as I said, here you can see an amino group, okay, NH2 or NH3 plus, and the right side you can see a carboxylic acid group, okay, that is COOH or COO minus, and here you can see a CH. This is hydrogen that is attached to this particular carbon, and this carbon is known as alpha carbon, and you can see an R group, and R group is nothing but the side chain. So this is the general structure of an amino acid. More clearly, R, CH, NH2, COOH, and this carbon atom that is present in the center, that is known as alpha carbon atom. So in this alpha carbon atom, one side chain is attached, NH2 group is attached, one hydrogen is attached, and one carboxylic acid group is attached. So that is what I told you, alpha amino substituted carboxylic acid. Okay, so these are amino acids. Now, as you all know, there are 20 different amino acids that constitute the formation of the protein, and the 20 different amino acids are, their naming is done by 3 approach. Okay, one is the full name. Okay, that's full name of the amino acid. Next one is the 3-letter abbreviation of the amino acid, or this is very important. And another one is a single letter code of amino acid. Okay, and this the full name and 3-letter and the single letter, and these are obtained from genetic code. There are 64 codon, they code for various amino acids. Okay, and these 3-letter codes are very important in bioinformatics studies. Okay, and the amino acids are alanine, arginine, aspargine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, and valley. Okay, so these are the different types, and this color indicates some of the amino acids comes under the same family. Okay, that is why. Okay, and here alanine is ALA, that is single letter code is A. Arginine, the 3-letter code is R, ARG, and single letter code is R. Asparagine, that's ASN, the single letter code is N. Okay, aspartic acid, ASP, single letter code is D. And cysteine, this is CYS, single letter code is C. And glutamic acid, GLU, the single letter code is E. Glutamine, the 3-letter code is GLN, and that is single letter code is Q. And glycine, the 3-letter code is GLY, and the single letter code is G. Histidine, this 3-letter code is HIS, and single letter code is H. Isoleucine, that is ILE, that is I, single letter code is I. Here you can see leucine, LEU, the 3-letter code, L is the single letter code. Lysine, LYS, okay, is the 3-letter code, and K is the single letter code. Methionine, MET is the 3-letter code, and M is the single letter code. And phenylalanine, PHE is the 3-letter code, and F is the single letter code. Okay, proline, PRO is the 3-letter code, and P is the single letter code. Serine, SER is the 3-letter code, and S is the single letter code. And threonine, THR is the 3-letter code, and T is the single letter code. And tryptophan, TRP is the 3-letter code, and W is the single letter code. And tyrosine, TYR is the 3-letter code, and Y is the single letter code. And finally valine, VAL is the 3-letter code, and V is the single letter code. So this is, you need to study, you need to by heart. This is very, very important for studying the advanced aspects of the sequence of protein. This is very, very important. So that is what I told you. Whenever you wanted to do any bioinformatics studies, these 3 single letter codes are very, very important. So these are the 20 different amino acids, and these 20 different amino acids are the primary amino acids in the genetic code. Okay, and these are the structures of this single and their molecular formulas and their structure. The simplest amino acid is known as glycine. Okay, and then it is getting complex, and you can see the complex amino acids. Okay, there are some of the amino acids are sulfur containing amino acid, that is methionine and cysteine. Okay, and methionine and cysteine. Okay, so these are sulfur containing amino acid. Okay, some of the amino acids are aromatic amino acid, that is tryptophan, tyrosine, and phenylalanine. There you can see aromatic rings. Okay, and this histidine as well. Okay, so these are the aromatic amino acid, and there are similarly lot of classifications are there, and this, if it is possible, the structure also you have to study. Okay, and we will discuss the classification of amino acid later in this unit. And the another important aspects of the amino acids are the major features of amino acid. The first one is the optical activity. The same thing we have discussed in the case of carbohydrate also. So the amino acid, the first carbon atom is the part of the carboxyl group, and the second, okay, the first carbon atom is the part of the carboxyl group, and the second carbon atom in which attached to an amino group, that is known as alpha carbon. I told you this one. The alpha carbon of the most of the amino acid is joined by covalent bond to 4 different group. Okay, COOH, H, NH2, and side chain. Okay, and that is the 4 different amino acids, 4 different groups. Okay, and this alpha carbon in all the amino acids is asymmetric or chiral, except in the case of a simplest amino acid called glycine. Okay, they are not same. Okay, here you can see there are 2 hydrogen here. Okay, and they are asymmetric, and the simplest amino acid, except the simplest amino acid called glycine, and where this is symmetric. Okay, and all other amino acid, this is a chiral carbon atom. Here you can see this 1, 2, 3 or 4. The carbon atoms are satisfied. The 4 different valencies of the carbon atoms are satisfied by 4 different groups of atoms. Such kind of compounds are known as asymmetric compounds or asymmetric carbon atom or chiral carbon atom. We have already discussed. So due to this chiral carbon atom or asymmetric features, the amino acid except glycine can exist in 2 optical active forms. This is very, very important. Okay. So one particular optical isomeric form, the NH2 group in the right hand side like this, and this form is known as D variety or D form, as we have discussed in the case of carbohydrate. And if the NH2 groups are in the left hand side like this, that is L amino acid or L form. So this is all about the optical activity. So amino acids are optically active. They can possess both the D and L variety. But these L varieties are most important than the D variety, which are occurring in the in all the protein in the natural form. And next one is the specific rotation. So the amino acids also having a particular rotation with respect to the plane polarized light. So as I said, the amino acid found in the protein that is belongs to the L series, many of naturally occurring L amino acid rotate the plane polarized light to the left. That is why it is known as levo-rotatory. But the other amino acids, okay, the other form of the amino acid that rotate with respect to the plane polarized light to the right, okay, that is dextro-rotatory, okay. And you can see this we will represent L and D. But in the case of amino acid, we generally don't use this L and D always. There is another representation called R and S that we will discuss later. Okay, so this is the specific rotation of some of the amino acid. Okay, all the forms are L variety. I told you that is most powerful amino acid that has a specific rotatory power against plane polarized light. Okay, the L-alanine, the specific rotation is minus 86.2. L-histidine, it has got a minus 38.5. Okay, L-phenylalanine minus 34.5. Then threonine, L-threonine that has got a negative 28.5. Okay, L-serine that has got a 7.5. L-alanine that has got a positive 1.8. And L-glutamic acid plus 12. And L-isoleucine plus 12.4. And L-arginine at 12.5 and L-lysine 13.5. Here is full of negative. In this table, L-lysine has the least specific rotatory power and L-alanine has the least specific rotatory power and the L-lysine has the maximum specific rotatory power. So this is the next important feature of the amino acid. Then the next one is the physical properties of amino acids. Okay, so generally these amino acids are colorless and they are crystalline substance. Okay, they are crystalline and colorless, and the crystal form may vary from slender needless, such as amino acid like tyrosine that shows a slender needless crystal, to a thick hexagonal plate. Okay, cysteine shows a thick hexagonal plate. And this amino acid, there are some amino acids are tasteless, such as tyrosine. Some amino acids are sweet, such as glycine and alanine. Some amino acids are even bitter, such as arginine. Okay, and the specific rotatory group, both the L or D variety or S and R variety, there have a different property. So that is why this specific rotatory power is very, very important. With respect to the rotatory power, they have got a different features. Okay, so say for example, S-phenylalanine is bitter in taste, whereas its enantiomers, the R-phenylalanine, is sweet. Just opposite. That is what I told you. That is the just with respect to the enantiomeric form has got a lot of differences in the physical property. and the amino acids are got generally high melting point, approximately 200 and above. Okay, these are soluble in polar solvents such as water and ethanol, and are insoluble in non-polar solvent such as benzene or ether. Okay, so this is another important property. Then certain amino acid, they can absorb the ultraviolet rays at 260 to 280 to 90 micrometer, the nanometer, that is such as tryptophan, tyrosine, histidine and phenyl, and they are able to absorb ultraviolet rays. Then as I said in the previous in the beginning, these 20 amino acids or the proteins are referred to as the standard primary normal amino acid. Okay, these 20 amino acids are referred to as standard primary normal amino acids. Then the next important feature of amino acid is electrochemical properties. So this amino acid has got both acid and basic properties. Okay, so I told you one end of the amino acid is an acidic group, another end is a basic group. Thus it has got an acidic and basic property. Okay, so the amino acid react with both acid and base, so thus they are amphoteric in nature. Okay, so what are amphoteric component? Any component they are able to react with both acid and bases are known as amphoteric component. So in the form of iron, the proton migrate from the carboxyl group to the amino group and produces the carboxylate, okay, and ammonium ion. This is very important. Okay, and in general case, what will happen? CHCOO, H and NH2. This is the simplest amino acid called glycine. And what will happen? Normal condition, the certain specific physiological condition, what will happen? The proton, the proton from the carboxylic acid group, the H will be migrate and bind to the amino group. That's what will happen. The COO become minus, and this will get a new proton and it will become NH3 plus. Okay, HCH, COO minus and NH3 plus. And you can see this particular molecule, one end of the molecule has got a positive charge and another end of the molecule has got a negative charge. This is very important. Such kind of a molecule is known as sweeter ion. So one of the most important property of amino acid, they are able to exhibit in the dipolar state with both the charge. Okay, such kind of components are known as sweeter ion. Okay, so this carbon atom is known as alpha carbon atom, that is alpha carboxylic acid and alpha amino group of the amino acids are ionized in a solution at a physiological pH with the deprotonated carboxyl group. Okay, the proton is migrated from the carboxyl group and that has got a negative charge, and that has got a negative charge, bearing a negative charge, and the protonated amino group, they are getting this deprotonation, removal of proton. Okay, from this case, this it is a deprotonation, and here it is a getting a proton, that is protonation to the amino group. Okay, NH3 plus, thus it will be getting a positive charge. Okay, such kind of a dipolar state. Okay, due to this, here it has got a positive charge, here it has got a negative charge. Okay, so that is why this is known as sweeter ion of glycine. Okay, so that is another important aspect. So amino acids are able to do electrochemical exchange, the positive and negative charge they will be able to exchange, and they are able to exist in the form of both the positively and negatively charged ion or both the ion. Okay, the compound has both the ions ionic form in the same structure. That is very important. So these are the basics about amino acids. Then next one, proteins. The proteins, the name protein is suggested in 18th century, 1838, by a Swedish chemist called John Jacob Berzelius. Okay, so he is the first person he coined the term protein. Okay, later another Dutch scientist called Mulder, okay, he defined the protein. Okay, proteins are the complex organic nitrogenous substance found in the cell of the living organism. Okay, proteins are the complex organic nitrogenous substance found in the cell of the living organism. These proteins are the building blocks of the body. In all the body parts of all the organisms are made up of proteins. And these are large biomolecule. They are macromolecule which consist of one or more chain of amino acids. Okay, one or more chain of amino acids. And this protein can perform vast variety of function within the organism, such as catalyzing the metabolic reaction. All metabolic reactions are catalyzed by protein, that are enzymes, replication of DNA, then responding to the stimulus, the signal transduction, providing structure to the cell and the organisms, and providing the transport of the molecule. Okay, and they do the transport of the molecule from one location to another location. So the proteins are arises from the DNA. That is why we have discussed that before this particular unit we have discussed nucleic acid. Okay, the proteins differ from one another primarily the sequence of amino acids. Okay, sequence of amino acid mainly in the side chain. Okay, so here the amino acid mainly in the side chain, okay, which is detected by the nucleotide sequence of their genes, okay, and which usually result in the protein folding into specific 3D structure that determine the function. Okay, so the protein it perform the function, and the function required a characteristic a three-dimensional structure. That three-dimensional structure is the responsible reason for the characteristic function, and that 3D structure will be produced during a particular event called protein folding that we will discuss in one of the topic in this unit. Okay, then as I said, there are total 20 different amino acids exist in protein, and hundreds to thousands of these amino acids are attached to each other in a long chain to form a proteins. Okay, and the proteins are the linear chain of amino acid which are called polypeptides. Okay, proteins are polypeptides and DNA is polynucleotides. Okay, don't confuse, proteins are polypeptides, DNA are polynucleotides. Okay, a protein contain at least one long polypeptide. Some polypeptide will be very short. Such a type of polypeptide may consist of 20 to 30 amino acid residues. Okay, that is short form, that is known as peptides, or sometimes known as oligopeptides. Okay, don't confuse oligosaccharides and oligopeptides. Oligosaccharides are the polysaccharide, the carbohydrate that consisting of 2 to 9 monomeric unit. Okay, but in the case of oligopeptides, they are short peptide, approximately 20 to 30 residues in length. And the individual amino acid residues are bonded together by peptide bond. This is very, very important. We will discuss. Okay, and adjacent amino acid residue. Okay, so all the protein has the structure. Okay, one end of the structure they have got an amino terminal end, another end of the structure they have got carboxylic acid group. A carboxylic acid group of one amino acid will be interacted with the amino terminal end of the another amino acid. Then there will be formation of a strong covalent bond will take place, that is known as peptide linkage. Okay, that we will discuss. Okay, and this individual amino acid residues are bonded together by peptide bond and adjacent amino acid with adjacent amino acid residues. And this sequence of amino acid residues in a protein is defined by the sequence of a gene. Okay, the sequence of amino acid in a particular protein, that is came from the sequence of gene which encode the genetic code. Okay, so there are 64 different codons. They code for 20 different amino acid. Multiple codon can code for same amino acid. This property is known as degeneracy of genetic code. We will see in the next semester in molecular biology. Then what are the major functional role of the protein? Why proteins are very important? Many proteins are enzymes that can catalyze the biochemical reactions, okay, and are vital to the metabolism, okay. They are able to catalyze many biochemical reaction. They are very vital to the most of the metabolic reactions, okay. And the proteins have a structural or mechanical function. As you can see, actin and myosin that is present in the muscle, okay, and the cytoskeleton, you know that microtubules are present, okay, and they forms a system scaffolding that gives the cell shape, okay. The protein can able to produce the cell shape and the integrity and rigidity to the organism. And the proteins are important in cell signaling, the passage of the signal, then immune response and adhesion of the cell, okay, cell communication and cell cycle, okay. And in organism including human, proteins are needed in our body. Out of 20 protein, 8 proteins are known as essential. 8 amino acids are out of 20 amino acid, 8 amino acids are known as essential amino acid. The remaining 12 are non-essential amino acid. What are essential amino acid? They are essential to the body. In our body, they are not able to synthesize. That need to be supplied through the diet by the food item, okay, that we cannot synthesize. And this protein, how will you separate the protein? There are a lot of techniques can be used to separate the protein. The protein may be purified or separated or quantified by other cellular component by certain techniques known as ultracentrifugation or precipitation, electrophoresis and chromatography. These are the major techniques in which we can segregate or separate, quantify and identify the different proteins. In your syllabus, they have mentioned electrophoresis that we will discuss. And I told you in this unit the very important aspects in biochemistry is to study the structure of the proteins, okay. How will you study the structure of the protein? There are so many methods. Why do you want to study the structure of protein? Then only we will be able to study the function of the protein. There are so many methods in which the protein structure can be studied. The most important and well studied example is X-ray crystallography, then nuclear magnetic resonance or NMR, then next one is the electron microscopy, okay. The latest technique is cryo-electron microscopy. In 2017, the scientific community got a Nobel Prize. A scientist called Joachim Frank, okay, along with another scientist, okay, Henderson, he got a Nobel Prize, okay. Then some sense mass spectroscopy also can be used to study the protein structure, and site-directed mutagenesis can be used, and immunohistochemistry also can be used to study different proteins. So these are the various methods to study the structure of the proteins. Then the next topic illustrated in your syllabus is important function of the protein. They have mentioned a brief idea, okay. This is a protein is the central, you know that we have discussed. Protein can produce the structure and mechanics such as actin, myosin, troponin, etc. They can act as they can catalyze the biological reaction, this enzyme, trypsin, pepsin, DNA polymerase, etc. They can act as hormones, glucagon, insulin, gastrin, etc. The protein can act as a signaling molecule, okay, adenylate cyclase or annexin, etc. Protein can act as antibodies. We will discuss immunoglobulins, okay, in our bodies. Then protein can give the fluid balance, okay, the liquid fluid balance in our body, that is mainly albumin and vasopressin. Then the acid base balance, protein can maintain the acid base balance such as plasma proteins. Then protein can act as the canals and pumps, okay. They are able to perform the transport, okay. That is the band 3 protein, sodium, potassium, ATPase. These are membrane protein. They are able to act as the channels and pumps. They are able to pump, okay. The next one, they are able to transport, okay, GLUT, rhodopsin and albumin. All such kind of protein are the most important protein acting the transport function. These are a very brief outline. Then little bit detail. So whatever we have told, once again we can discuss. Many protein can act as the catalyst, such as enzyme. They are biological catalyst. Most of the biochemical pathways, glycolysis, Krebs cycle, okay, all those pathways will be mediated by various enzymes. Then the protein can be act as a structural compound. Say for example, fibrous protein can serve as a components of the tissue which hold the skeletal elements. Okay, collagen is a structural compound in connective tissue. Then similarly, nucleoprotein can serve as the carriers of genetic material, genetic character, and that governs the inheritance of traits, okay, from organism to organism. It will transmit. Then protein can perform transport function, as I said. Then protein can act as the hormone, as I said. So many hormones protein can produce. They can act as a hormones. Then this can regulate the growth of plants and animals, and they can control several physiological function in our body. Okay, under certain condition, that is under the condition of non-digestion or no chances for the denaturation, in the absence of denaturation or in the absence of digestion, the protein will accumulate in the body and they produce the toxicity. They contribute the toxicity. So if the protein is not denatured or not digested properly, then it will create a problem. That is why you have studied the digestion and absorption of protein in physiology. That is very, very important. Okay, then certain protein like blood plasma, okay, that means to say if you centrifuge the blood, you will be getting if you remove the blood cells, you will be getting a straw colored fluid, that is known as blood plasma. That is very much essential solution that contains lot of antibodies. Okay, essential a solution of proteins. Okay, that proteins are mainly antibodies. Okay, there are certain glycoprotein called interferon. They are very, very important. Okay, these are low molecular weight regulatory glycoprotein produced by several eukaryotic cell with respect to numerous inducers such as a viral infection. Okay, then double-stranded DNA, endotoxin, antigenic stimulus, mitogenic agents, and many parasitic organisms capable to grow intracellularly with respect to this. Okay, and immediately the body will produce interferon that indicate the invading of all these agents. Okay, so there are certain protein that will represent. Then certain proteins called that is peptides, small short peptides called defensins. They are found to be antibiotic in nature. What are antibiotic? They are having the inhibitory property against other organisms. They are antibiotic in nature. Then certain types of peptide called endorphins, which are found in brain, which are involved in the suppression of pain and provide a feeling of joy. We will feel happy, and that will be, if you introduce this particular peptide, what will happen? That will be contributing suppression of pain. Sudden, that is a pain a suppressor. Okay, these are some of the important function as I have said here. So these are the overview of the function. Okay, so this is all important functions of proteins. Okay, so this is all about amino acids, proteins, and their function, dear students. So we can conclude today's class. Once again, we can glance through the main points studied in this particular class. Okay, we have started the amino acid. There is a brief introduction we have made. There are 20 amino acids. How they are naming their 3-letter code, the common name, 3-letter code, and a single letter code. That is very, very important. The structures of various amino acids. Okay, sulfur containing amino acid, their classification we have not discussed in detail, but the structural aspect can give you lot of insight. Then certain peculiar properties of amino acids as optical activity. They can produce both the D and the L variety, but L varieties are common. Okay, then specific rotation. They are able to rotate with respect to plane polarized light. Okay, their physical property, the lot of physical properties. The electrochemical property, they can exist as they can ionize, and they can exist as both positively and negatively charged component in the same structure, that is known as sweeter ion. They can exist as sweeter ion. They can ionize. Then we have discussed the proteins. Okay, the amino acids are the basic building blocks of the protein. Then we have discussed the fundamentals of the protein. and why the proteins are very important, and what are the important functions of protein. So these are the important points we have discussed in this particular class. So these are the major textbook, dear students. You can refer Biochemistry by J.L. Jain and Principles of Biochemistry by Leninchar, the best textbook, and all this textbook also you can refer. Okay, thank you dear student for watching. If you are having any clarification, you can mail me, or you can write or comment below so that it will be answered, or you can contact me in person. So thank you very much for watching this particular class.